Alright, can we get an audio check? Alright, we're good in an audio. So hello everyone, my name is David Rock. I'm here at the University of Maryland in Baltimore, and welcome back to Cytometry in R. So today is going to be one of the extra bonus walkthroughs that accompany the week 12 to 14 sections, namely focusing in on the spectral signatures. So today in the extended bonus walkthrough, we're going to basically go ahead and see how to retrieve out, not like we saw last week when we were retrieving just a single fluorophore signature from APC Fire A10. We're going to see more on the complicated angle of how do we assemble this for the 20, 30, 40, up to 60 spectral fluorophores that you might have in any given panel. So, during the weeks 12 to 14 of the Cytometry in R course, we are primarily focused on how to work with raw spectral flow cytometry files. So, during week 12 that we just went through, we learned how to extract out and characterize fluorescent signatures that are present within our mixing controls, both the single color and the unstained. This we will follow up next week where we're going to compare these signatures to each other, allowing us to predict how they will interact in the context of a full spectral flow cytometry panel. And then finally, in week 14, we're going to take those fluorescent signatures, which we're going to derive today, and then use them to basically go ahead and unmix our full stain samples, bringing everything full circle. So, due to Cytometry in R's use of GitHub, we're not able to include full-sized FCS files as part of our actual dataset, given that many of these in their original form are easily over 100 megabytes each. So consequently, for the datasets that were included as part of the week 12 and week 14 materials, we ended up downsampling them rather substantially to be able to get around this issue. However, working with a complete dataset can provide you additional insight, because it oftentimes will mirror that same experience that you would be doing with your own dataset. So this particular bonus walkthrough is particularly focused on the steps that are going to allow you to retrieve and process your original FCS files, very much in a similar way to what I did originally for that import repository dataset we ended up grabbing. So, in the journey so far, back last night in the setup for signature retrieval walkthrough, we ended up transforming, gating, and then verifying the gate placement for each of our individual unmixing control gating set objects. These included both a single color gating set for cells, a single color gating set for beads, as well as a gating set for just the cell unstains. And then ultimately what we left off at the end of that walkthrough was we saved each of these as its own intermediate.gs object, which we are then able to now come back in and reload back in without needing to redraw and readjust all the gates, expending additional effort to do what we've already done. So we're going to pick right back up with these intermediates, and then go ahead and work towards retrieving fluorescent signatures for each of our single color controls so that we can derive out a signature matrix. This we then can use next week when it comes to doing our comparisons for week 13, but then also during week 14 when it comes to unmixing the full stain samples. So this walkthrough will share a lot of content with what we've already covered in week 12. However, the week 12 walkthrough is very much more granularly focused on explaining how to go about the individual signature extraction. It's in the hopes of being able to break open what for many in the community is often essentially a black box. This walkthrough is assuming you already covered or at least skimmed some of those contents, so if you're interested in like the little tiny mechanistic details of each line of code, that would have been covered. But this walkthrough is more focused on providing a more faster, efficient method via which we could scale to the level of our experiments thereby being able to get the signature matrices that we need, very much more towards the experience we would see for commercial software. But also while providing the launching points for delving further when it comes to aggregating single-cell normalized signatures that are going to be the focus of the next bonus walkthrough. So, a couple bits of housekeeping. As we mentioned already in the previous walkthrough, we are currently using the developmental version of the flowGate package that contains several new functions that allowed us to adjust the individual gates as far as their placement on individual specimens. So, due to the twice-yearly release cycle of the bioconductor packages, the easiest way to take advantage of these new functions is if you go ahead and uninstall flowGate and then reinstall it from the GitHub repository. But for those of you who are currently looking at the discussion forums, you can see there's been an issue recently where there has been some additional changes made behind the scenes to the flow core, the flow workspace, the ggcyto package, and how they're able to handle the transform functions. And that behavior has not yet been reflected when it comes to the flowGate package. So at this point in time, if you do go ahead and reinstall either flowGate or Luciernaga, which we're going to do in the next couple slides, if you get to that little screen right as you start to download the package where you ask you, do you want to update all packages, some packages, or just the packages from CRAN? For now, until those bug fixes get implemented into flowGate, just say don't update any additional packages, or select only update the CRAN packages until the maintainers have had time to actually implement the fix for the change in behavior for the transform. So as you encountered at least a couple times in this course, any change that's happening within an open piece of software has the potential to impact all the other dependencies, and it's just how do we navigate them as we encounter these changes. So, if you have already done this as part of the last walkthrough, you're all set. However, otherwise, if you need to go ahead and re-update flowGate, the way you're going to be able to do this is first remove the R package, close and reopen positron, and then go ahead and install a new version. So you can see here, we do use the remove.packages function to remove flowGate, and then once we had reopened positron, we could then run remotes double colon install underscore GitHub to then go and download it from GitHub itself. In which case, we're going to the original GitHub account, in this case NKInstinct, / the repository, which in this case is going to be flowGate. So once that's done, we can go ahead and just make sure we got the correct version installed, which we're going to do via the package version function. And to be having the version that you need to be able to do this particular walkthrough, it needs to be at least 1.31.1 as of right now in July of 2026. You need to be at this version or something greater when it comes to what we're able to do. So additionally, there was also some additional changes that were made to the Luciernaga package. Namely, a lot of what we will be doing today is those new orchestration functions that are intended to setting up the floor for specific gates that would allow you to correct for the single colors way easier to do when it comes to specifying those gates individually, which was the old way of doing this. So we'll also need to install the updates for the Luciernaga package. So if you run the package version on the actual Luciernaga chunk, it should be at least 0.99.10 or a greater version than what we currently have to be able to get to these orchestration functions. So we can check it once again using package version. And if not, if you're something before that point, you just need to go ahead and uninstall it, reinstall it, similar manner as what we saw for flowGate. In this case, we're just changing the GitHub account is under my GitHub account, and then it would be / Luciernaga in terms of the repository. So, at that point, having gone through a little bit of the housekeeping and verified that those two correct package versions are being installed, we can then go ahead and proceed and attach everything we're going to need for today in our local environment via the library call. And what we're going to load for today for now is just going to be flow workspace, flowGate, dplyr, purrr, and stringer from the tidyverse packages, and then Luciernaga, just to make sure they're all active in their environment and that we have access to the functions that they contain. And then once that's done, we just need to do a few of the housekeeping steps we'd have encountered in one of the other walkthroughs. First off, we need to respecify the file paths to both our storage and output locations. So in this case, I'm specifying my storage location based on how I had organized my folder. It's going to be within data folder, within the SDY3080 folder, where all my little individual FCS files are being stored. And then my output location for anything intermediates that gets stored, I'm just going to set that to the outputs folder, which is located directly within my working directory. And next up, given that not everybody's workstations will be set up in the same way, I do recommend working with each of these gating sets separately, so one at a time instead of loading them all in at once. This should help you reduce the total amount of RAM that might be taken up at any given point in time, giving you more chances of having the code succeed. So let's go ahead and start first with the bead single color unmixing controls, because theoretically these should be the easier examples to be able to work with, because there's going to be substantially less autofluorescence to handle. So to get started, let's go ahead and reload the gating set from where we had stored it in the last walkthrough in the form of its intermediate.gs file. So for easier handling, we are going to save it under the exact same name we used during the last walkthrough, just so that we avoid having any confusion. So at that point where we had left off, we saved it within this bead single colors _gs folder, which we had appended the .gs annotation for. So we just respecified a file path to where we had stored it, and in this case we can use the flow workspace package load _gs to load it back into our active environment. And at this point we're just assigning it back to what it had been saved as in our local environment before we ended up saving it to the intermediate folder. And then at that point, we just need to make sure that all the specimens we are expecting to be there are still there, and all the gates that we had assigned are also still there. So if I run pdata, I can see that, yep, all my bead single color controls remain in the gating set just as we had left them, which is encouraging. And then at that point, we can start considering all the other little bits and pieces. So from what we currently have in the metadata, we can see that we have bead single color controls, these that had been acquired as part of the IoT_00 experiment. So when we were checking through the methods / the metadata that was provided for the SDY3080 study, or alternatively, if you don't have access to the metadata and you're just trying to reverse engineer it from internal keywords, we can find that all these individual files were originally acquired on a Cytec Aurora, one of those 5 laser configurations. So currently within that file name, we have several bits of information that would be rather useful if we're able to extract them out and place them in their own column. So either through the use of the pdata or just as a standalone in terms of keeping them as a data frame or a tibble object. So from that same set of file names, we can see they contain a fluorophore, they contain the antigen, and they will also contain information about that particular control type, in this case beads. So seeing as currently everything's just being stored within the column name, it would be quite useful if we're able to repeat the appending metadata steps that we have encountered in other walkthroughs throughout the last couple weeks, so that we just need to extract out the bits of information we need and create new columns. And we can do this using the stringer package, especially doing structure detect, structure extract, and then just need to combine it with a little bit of those regular expression, i.e. the regex code snippets, so that we're actually grabbing the parts that we want from the original file name. So as we were covering back last week of the class, you can do this using a large language model like an LLM, so ChatGPT, Claude, whichever your AI go-to would be. And you can ask it a very specific question of, I have this particular file name I'm working with in R. I'm trying to grab these specific portions and do it consistently across multiple names. And it will spit back this kind of code here that you can use to actually extract it out. And then at that point, it's, of course, still a LLM output. It may or may not work, but you can at least test it with your code. If it does get you back the fluorophores, the antigens, and the type, then you're all set to go. If it's not, and it's really a large garbage pile, you can then try again or go and look elsewhere as far as the information goes. But ultimately, we had encountered the same line of codes back during week 12, so we know the end result. We were able to correctly extract out both the antigen, the fluorophore, and the type from the overall name that's present within the metadata, and we could update them as their own individual columns. So what this is going to look like once we had done that initial updating, you can see we still have that row names present here. Then we have the actual name column. We can see we have that fluorophore name column, the antigen name column, as well as the type column here. So, as we were encountering back during week 12, as we are skimming this output, we do notice there's a few places that the extraction did not work quite accurately. Namely, for the first sample, which was named 4 beads unstained, it wasn't able to find that same kind of syntax when it came time for structure extract, so all those values came back showing up as NA. And then, though, we have a similar situation where the original type of naming convention kept on our Cytec Aurora at our shared resource facility had slightly different naming conventions in terms of the p-vial 770 and p-dazzle 594, at least in terms of not having a space versus actually having a space between vial 770 and dazzle 594. So kind of similar to what we deep dive for the week 12 material, we can go ahead and edit all these individually in place programmatically from R using the mutate function in conjunction with the case when function, both of these being from the dplyr package, to basically do little point edits to these particular file names. So what that looked like in practice, basically for the bead unstained, we're just rechanging it to be NA for character in case of its antigen. And then for the fluorophore column, we're just having a case when situation where for when the column name contains a value that has bead unstained in it, we end up renaming the fluorophore for that particular row as bead unstained. And then same thing, we were doing little point corrections. So when we're checking within the fluorophore column and you encounter within the fluorophore column p-vial 770 without the space, we convert it to p-vial 770 with the space. And same thing for p-dazzle 594 without the space would be converted over to p-dazzle 594 with space. So that at that point, after these little point edits have been introduced, you can see this is kind of really blurred out, so it's kind of hard to see from this view, but you can see right around here, p-vial 770, p-dazzle 594 now have the space in the correct location, and now instead of na across the board for bead unstained, we can see that the fluorophore has a value called bead unstained as we had designated. So with these adjustments now being made, we're able to basically overwrite the existing metadata for our gating set and replace it with our own variant that will contain these additional columns that we can then use if we decide we want to subset down the data further. So kind of similar to what we've already done, we're just, through the use of our assignment arrow, overwriting the current contents of P data for our particular gating set, and once they're overwritten, if we run it, we'll see or get the correct type of metadata structured out. So as we proceed, with the metadata that we have now updated, let's start stepping back and consider some important elements that are still missing that we didn't quite cover as extensively last week and during week 12. So currently, our single color unmixing controls are present in a relatively generic gating set. The last common gate that they all have in common with each other is the scatter gate. So from here, based on the per individual single color control, ultimately, if we're trying to extract out a signature, we're going to want to create an individual gate based off of that peak detector for a given fluorophore being in the x-axis position. We will also want to make sure that we're able to adjust the individual gate that is created in order to accord with the best practices that we as a field have come up with when it comes to single color controls, so that at which point, when we're happy with those individually gates that are placed for the peak detectors of each fluorophore, we can then extract out the cells or the beads that are contained within that gate, and then do very similar steps to what we saw last time with week 12 material to actually subtract out the background, derive the actual signature, so that we are able to use it. So, in addition to not only designating out what's going to be on x-axis, we would also need to designate from what thing is being used to subtract out the background, especially in the context of if there's multiple types of unstains that you're working with. So let's first start off by retrieving the metadata. Let's go ahead and save this as an intermediate data frame. It's a little bit easier to work with a data frame within our working environments rather than messing around with the pdata internals directly. So once again, grabbing the metadata that we had just updated, placing it within this object template. So, using the dplyr packages mutate function, let's just go ahead and create two new columns right now. We can create one for detector, which we can leave empty for now by just putting two quotation marks right next to each other. And let's also create a negative column, and let's provide it the value bead or unstained, since currently for this bead gating set there's only a single unstained, and that is what we are calling it in terms of the particular fluorophore column. So how do we actually go about and do this? Well, kind of similar to what we encountered in the past. We're taking a little data frame object, we're piping it through to the mutate function, and then we're creating 2 new columns since they're not already existing. Detector is equal to, between the little quotation marks, there's nothing, so it's going to be left empty. And then same thing for negative being a new column, we're setting everything equal to bead unstained in terms of the values. At which point, if we visualize it, and I know this is pretty zoomed out, but we can see detector column looks pretty bare. There's no value actually being applied. But we have a new negative column specifying what is being subtracted out in terms of the background. We can see all across the board for all our specimens, it's showing up as bead unstained, at least for this particular gating set. So, since this data originally came from a 5-laser Cytek Aurora that has 64 detectors, and because the Luciernaga package was originally optimized on Cytek Aurora data, there's a good chance that the fluorophores that are being present for this particular panel will be actually covered in the database for the package in terms of fluorescent signatures. So, if we're trying to determine what the peak detector for each individual fluorophore is, and we haven't been doing flow cytometry for a while, we could leverage the Luciernaga package to try to make an educated guess about what the peak detector should be for each of the fluorophores that we have. So, the way this would basically work is we're mutating and changing our existing detector, and we're doing this through basically iterating through the individual fluorophore column contents. And this is one of those weirder syntax when it comes to iterating with a function. Just let's try at this point following what it's actually doing. There's a little function within Luciernaga called detector text, detector guess, and then at that point it's iterating in whatever's being assigned to this F object, in which case as we map, it's going to be whichever fluorophore happens to be in sequence. And then we're passing an internal argument. There's 64 detectors on a Cytek Aurora 5 laser, so all in all this is giving some little bit of context. So this is still not necessarily, for a beginner, the most interpretable code at the first glance, but let's see what it actually does in practice. So at this point, as we start seeing where the detector column had been empty, we're now coming up with guesses for what was actually being present. So as you can see, this is going to be BUV615. It's showing up as the UV10 detector. If I check for where's my APC Fire 810, because that's the one that's remaining consistent, that one's showing up as the R8 detector, which makes sense based on what I know about that fluorophore. So you can see the guess has come up with a result for most of these across the board, except for the 2 problem children that didn't have a space for this particular screenshot, which are showing up empty at this stage. So if you are one of those instruments that is covered, this is one way of being able to actually go ahead and provide information about on which detector do you want to build that x-axis gate for a corresponding fluorophore. But there's still a couple that you would need to edit in, either by writing this out as a CSV file or alternatively doing the mutate casewing conjunction to be able to do so. So this is a similar statement. So if you're working with the data from a different spectral flow cytometer that wasn't actually accounted for correctly within the Luciernaga package, you can always correct these either manually or by doing the mutate case1 function, depending on what you're trying to do. So what you could also do is just go ahead and double check. If you're doing this manually, you don't have any additional contact information. You just want to make sure you're providing the correct form of the syntax for what is actually being seen as a column name for your particular FCS file. So in this case, you can see checking this one, it's all area variants when it comes to the Cytek Auroras. You might have multiple variants when it comes to a Sony or a BD or a Opteon kind of style system. And then, of course, it's the statement I've already made a couple times. You can either write it out as a CSV and make your changes that way, or set up your code just to modify it programmatically via R. So if you were writing it out as a CSV, this would kind of be the same approach of saving your data frame to a new CSV file in whatever output location that you have specified. Alternatively, what we showed before for the mutate and case1. So if you're doing this as part of your regular workflow and you do output this as a CSV file, one word of caution of what I would recommend is every time you run a Quarto Markdown document or an R script, it has the potential to overwrite anything that you end up correcting by hand. So if you do output one of these CSV files and you do put in the effort to update those detector columns with the corresponding matching detector, save that intermediate.csv file with a new name, and then load in that new name CSV file into R. So that way, even if you do accidentally hit run all chunks above, the CSV file won't accidentally overwrite each other, and you'll still have the properly named formatted to be able to use. So in this case, you can see that original output was named bead match. I changed the name of the copy to updated bead match, which I would then be able to load back into R. And at this point, you can see this one still wasn't corrected for the screenshot, so this is one that needs to be updated accordingly. So when that's all said and done, you have an assembled template. It would contain most of the needed information in terms of creating a correct peak detector gate for each individual single color control. And then also subsequently, once we have verified those gates were created and placed properly, match the extracted values we get from those gates for background subtraction for whatever we've designated in the negative column, in this case the bead unstained. So before we go ahead and proceed, there's a couple things we should go ahead and double check, especially for this gating set. A lot of repository data often will have like duplicated single color controls. Many of these are because maybe you did not know originally if your original antigen fluorophore combination was going to be bright enough, so for those reasons you included an additional CD3 of that same fluorophore to be able to handle of that particular edge case. And vice versa, maybe you have two zombie controls just for one particular condition, or maybe something didn't work one day so you reuse the single color from the different day. All the different variations we would normally encounter during our regular flow cytometry experiments can actually come into play. But we don't want to save all of these different duplicates together. We just want to keep it to one single fluorophore represented once in the matrix when it comes time for unmixing. So if we want to check to see, is there actually a duplicate when it comes to our fluorophore group, what we can do is, this is kind of an example line of code you could run. You could take our data frame intermediate as we currently have it. We can use the dplyr packages group by function and just group together everything based on the fluorophore name. And then at that point, we can do a little bit of modification, mutating a new name count, which will basically have the value of row number. So if this got grouped based on FTSE, and let's say there was two FTSEs, they would come in with a data frame that's row number 1, row number 2. At which point, when you ungroup, you could basically filter out anything that has a value that's greater than 2. And then at that point, you can pull the fluorophore name and identify it out as the unique values. So in this case, we can see for this particular gating set in this kind of screening, we see that there's multiple Pacific Blue single color controls for beads, multiple FTSE single color control for beads, and multiple Alexa Fluor 647 single color controls. So we need to actually designate which of these we want kept and which of them we want removed from the gating set. So let's go see how to isolate them out and see what's up. So as we are actually running this, you can see we're grabbing our data frame and we're filtering for anything that has a fluorophore equal to those values that came up. So Pacific Blue, FTSE, and Alexa Fluor 647. So right off the bat, if we check for the 647s, we can see one of these seems to be a CD3 based 647. The other one is a V alpha 7.2, one of those markers that's used for mates. We can similarly see for Pacific Blue, we have a CD14, CD19, which are both part of that dump channel. So we just need to select the brighter of these two when it comes to generating our matrix. And then there's a similar story for FTSE. We have a CD3 FTSE as well as a V alpha 24, J alpha 18, which in humans would have been the NKT invariant antibody marker there. So we need to go ahead and basically select which of these we expect to be the brightest in terms of MFI, so that we are only retaining those when it comes time to generating our signature matrix. In the context of this kind of decisions, we know their CD14 monocytes are going to be a little bit on larger cells, so they may be a little bit more brighter across the board, but we can always go ahead and just quickly visualize the populations based on those two different versions of each other, identify which is actually the brighter one, and then select that one accordingly, which in this case is going to be the CD14. For the other two markers, which had a CD3 as well as the particular innate like T cell identity antibody, there's multiple options that you could do. So the reason there's these additional single colors based on those antibodies is for those rare cell populations, especially when it comes to single color controls, oftentimes if you have a CD3 of the same fluorophore, you can acquire that at the same time, and then worst case scenario, if you don't get enough events for the two ILT populations, you could substitute in the equivalent CD3 antigen with the same fluorophore kind of color. So we could use either of these, but generally what you will use for beads would be using the particular antigen that corresponds to what you're adding to your full stain sample, just so that you're matching across the board. So ultimately, if you went back into import metadata and verified with the paper, you would also see that the V-alpha-24, G-alpha-18 antibody, as well as the HCD1D tetramer, were not exactly on the same fluorophore. They were placed on FITC, and then the tetramer was placed on alexafluorophore 88. So there could additionally be one additional antibody that's present beyond the 29 colors that we're expecting. So ultimately, we just need to make sure these individual files get excluded when we create our gates, which can be done by filtering them out from the overall template. So as you can see, we had our intermediately assembled template, and basically, based off of that name column, we are removing the CD19 Pacific Blue variant, the two CD3s for 488, 647, and then that extra CD3 that was still present for beads at that point. At which point, we should be left with just the original fluorophores with no duplicates across the board. And at this point, if we rerun that line of code we encountered previously, no actual entries come up with the duplicated amounts. And if we scroll down now, we can also just make sure that, yeah, everything looks kind of good across the board. If we go down and check for p-Vial 770, p-Dazzle 594, we can see within our gating set, these have now been corrected. We can see for example, our APC Fire A10, which was our example for week 12, it's going to have a positive gate created based on the R8 detector, and then it's going to have the bead unstained background subtracted. And then similarly for BUV395, which is in the first couple detectors for an Aurora, it's on the UV2 detector, and it's going to have the bead unstained used when it comes time to subtract out the background. So this is what we have in terms of an assembled template. So at this point we have 29 rows, one that includes the unstained. The main reason for this is there's also Zombie IR, which is generally not used for staining the bead cell controls. It's generally going to be placed on your cell controls, which is something that you could just go ahead and check around to make sure it's actually proper as far as what you're expecting. Alright, and before we continue, let me take a sip of my coffee. Alright, so as we saw last week during week 12, even when we're working with a single fluorophore, there are a lot of moving pieces when it comes to extracting out a normalized fluorescent signature from an individual single color control. So combined with the fact that most spectral flow cytometry panels end up featuring way more than just one single fluorophore, we need the ability to scale up accordingly so that if we have any hopes of routinely unmixing our data in R, rather than just a little showpiece of, yeah, we did that once. So fortunately, now that flowGate has an easy mechanism by which we can create the span gates, the new version also has the ability to easily modify these gates once they are created, which will basically allow us to adjust them back and forth so that we're able to refine which set of cells get selected for. So in reality, the only really tricky component that remains is basically creating those individual peak detector gates that correspond to where we expect to see the peak signal for each corresponding unmixing control. So basically, to facilitate this process, within the Luciernaga package, I've created a new function, which is initial unmixed span gates, which when you provided this little kind of gating set, we provided our gating set, and we provided the completed template, it's just basically going to iterate through the process of creating these individual gates for you correctly on a per unmixing control basis. At that point, all you would really need to do in terms of the workflow is kind of similar to what you do in a commercial software. Open it with the flowGate package, visualize where the gate is, adjust it as need be, and then repeat for all the other fluorophore combinations. So, first off, let's go ahead and double check. Let's see what gates are currently present within our gating set. So if we use the plot function on our gating set, we get back this return. As you can see right now in the original state of this intermediate file, we only have a little singlets gate currently present for these beads as the last gate that's present. So since we are working with these beads, we're going to use that cleanup gate as the root that we end up adding additional gates to. And then also we need to specify some additional boundaries. Generally, I want to make sure I'm able to see them when I'm first getting started, especially if I'm planning to just adjust them all manually. So if we set them for like the 75th to the 80th percentile in terms of brightness, that should be somewhere in the middle of the range of the bright cells, and at which point we can then adjust them. So in terms of actually running this Luciernaga function, you're basically running the initial unmixed span gates, you're providing that template which contains the peak the detector column as well as the negative column. We're providing it the gating set that we want all those new gates to be added for. We're designating under which gate is going to be placed through this use of the subset argument. And then we're basically saying draw the lower bound at 0.75 or the 75th percentile in terms of brightness for that particular detector. And then the max percentile we can set at 0.99. And then at that point, we're going to hit run. And basically what's going to happen is Luciernaga is going to iterate through the creation of individual gates for each single color in our gating set. And what you're going to see is this little progress bar that's going to pop up that's just going to show you as it iterates how it's doing in actual production and actual duration as well as how far it's gotten so far. And then ultimately, once that progress bar reaches 100 and it finishes actually running the lines of code, what you're going to see at that point, if you plot your gating set, is something that looks like this, where however many different floors in your panel, there's an individual gate that has been drawn based on that peak detector corresponding to the name of the floor in question, and all placed directly below that gate of interest. So at that point, all the tedious part of the process of what would have been track down the APC fire's peak detector, find my single color for APC, create a new gate for the R8 detector on that single color, and then adjust it, and then repeat that process 29, 30, 40, 50, 60 times depending on the size of your panel. All that is just being automated away, and you really just need to check to make sure that the gates that were placed are actually accurate when it comes time for what you're trying to do to extract out the signatures. So, when we're going to actually go ahead and check to make sure these signatures are correct, we're basically running one new function from Luciernaga, which is the check unmixed span gates function, which has, let's go ahead and see how this works in terms of the basic mechanics before we just have it iterate through all the different ones we're going to see. So basically, this is a modified version of the GS apply gate check that we've encountered in flowGate last time. But in this case, it's just designating out the correct single color control specimen, as well as telling the equivalent gate that's being used to check at that same point in time. So let's see this in actual practice. If we are running this individually with the original flowGate code, we'd see it's GS apply gate check. We are going to be checking this gating set. We want to check this particular gate, and then the correct single color for APC Fire A10 would have been the 16th specimen in the gating set. So this is what we'd be doing if we were trying to check this individually within flowGate. At which point, it would just launch the Shiny app, we'd allow us to adjust it, and then save it, and then update accordingly for that specimen. So this is what this would look like in practice. It would launch again, show you where it had been drawn the gate in terms of the span gate. Because span gates are one-dimensional histograms, span is already automatically selected. So let's say you were not happy with this one, you wanted to make it smaller, you just draw a new span, select done when you're happy with it, and move on. So of course this plot can sometimes be off scale. You can try to rescale this by providing the approximate x value, which would go into the enable manual coordinates. For example, as you can see here, it clipped a little bit of the edge. So at this point, you could then modify your gate. If you reran that line of code and checked again, you would see it's actually a little bit abbreviated for what we have. So let's go ahead and double check that. If we are just checking to see, did it actually correct for that particular specimen, not just across the board for everybody? This would be one of the Flow workspace packages functions, which would be gs_popgetGate. And if we run that, you can see this is the Spark Blue gate. It's at 3096 in terms of its first span. If we check for FITC, it's at 396 as well. But we just made that correction to APC Fire A10 single color, and you can see the value is now changed from 350 compared to everybody else that was just kind of generically applied across the board. So yeah, we were able to actually correct for it in terms of just that individual specimen that we were actually interested in. So these coordinates have been adjusted, which is what we're trying to do. If we had been wanting to adjust across the board for everybody, we could have used that adjust all argument, which had been set to false, just to correct to the correct position for everybody across the board, which is what we'll explore a little bit more next time for the spillover comparisons actual run. All right, so let's put this in context. So far, we have our initial gating set. We used a couple of the new functions from Luciernaga just to make sure we actually created a template that could then be used to designate out the creation of all those new individual gates based off of the matching single color control with its peak detector when they're being created. And now what we really just need to do is something very similar to what we'd have for the commercial software, where we iterate through all these new gates as they are placed on the correct specimen, just to make sure when we're drawing a positive gate, it is placed correctly. So this is going to be what we're going to be running in the context of the check unmix span gates function. So what this is actually looking like is, once again, we're providing the template, which is what's being used to orchestrate everything together. We're providing it the gating set, and then at that point, we do want to go ahead and have it adjust everything to match whatever we set it for across the board for all the specimens. So at this point, we can see this is the very first gate. If we're happy with it, we could just select done. If we want to go ahead and make it as bright or brighter, or just even a little part of this brighter portion of the peak, once we have span selected, just draw a little span region, and then we'd be able to select done. And this is for BUV395, so at that point, we don't have to worry about BUV395 anymore. And then it would just immediately iterate to the next particular single color in our gating set. If we're happy with it, we could select done. At that point, it would iterate to the next one, same thing, same forth. We could just rapidly, in sequence, quickly adjust all these gates across the board based on the best practices as the field. So as you can see here, APC Fire 800, APC R700 was a little abbreviated for where we had been drawn in that initial gate. We can go ahead and draw it correctly in terms of the span, so we got as bright or brighter for everybody. So at that point, not only did we rapidly create the correct gates based on the peak detector for the correct single color controls that they match, but we also iterated through and double checked to make sure they were actually correctly placed. So at this point, we've done quite a bit of effort to get this gating set to the point we need it for signature extraction. So let's go ahead and save it as another intermediate GS file, just so that we have it in case we need it later on in the future. So in this case, I'm just going to go ahead and modify the original name of those intermediate folders. I'm going to say this one as _amplified in addition to the original name. Save that one out as its own intermediate file. So now I have both variants if I ever need to adjust either one of them. So at this point, we have created the peak detector gates, and we've adjusted them on the basis of their respective single color unmixing controls. So now we just need to retrieve the cells that are present in those gates, reverse the transformation, and then take the average measurement across each detector and get back the combination of basically that fluor and autofluorescent signature, to which we had been referring to as the pre-process signature last time around. So as we saw back during week 12, individually doing this can be relatively simple. However, remember, we have currently about 30 fluorophores for this panel, and your panels may be anywhere from 10 all the way up to 60 fluorophores. So we want to make sure that we're not regretting our life decisions at the end of going through all these different fluorophores and subtracting out the background. So in order to try to keep us sane a little bit longer when it comes to flow cytometry, Luciernaga has a wrapper function. Basically, it's the retrieve average signature function, which would manage all these steps on a per row basis for you, so you don't have to worry about grabbing the correct negative for everybody or grabbing the correct positive across the board. So what this looks like in actual run practice is same thing. It's a similar kind of syntax in terms of its arguments. We're providing it our assembled template. We're providing it our gating set. We're letting it know, yes, we do want to transform. And then at that point, it does its run. You get back this updated template, which will now have, in addition to what we had assembled being placed over here, we now have all those individual retrieved median signature values on a per detector basis appended on as additional columns as we go across the board. So the one thing we need to still remember is this at this point is just the average values that's seen on the detector. It contains both the fluorophore as well as the background. We would still need to mediate out the autofluorescence subtraction, which is where that negative column that we ended up generating, which contains the name of the respective specimen, in this case bead unstained, is going to come in and become part of what we need to be able to do. So basically, if we check right now for bead unstained, if we ran the expers iterated function, we could retrieve out what the actual signature of this bead unstained is going to look like. And this would basically be our signature of the bead unstained, kind of similar to what we've encountered in the past couple weeks. So we have now basically what the signature of our unstained specimen is. But if we go back to what we had for that retrieved value, you can see it wasn't able to correctly retrieve out the actual signature from the bead unstained just because no detector value was provided, so we had a little breakdown in terms of the code. So basically what we need is we have the signature values for our bead unstained. We need to basically substitute them in for this particular NA that's all across this individual row. So we basically need to cut and paste it in terms of R. So once we are able to grab that same equivalent information for our bead unstained, we need to make sure it gets added back into that updated template just to make sure everything is restored to how it should look. And as you can see, if you look at the column names for our template, we can see the first several are all that little bit of metadata, but the first actual numeric value we need to be using is going to be, looks like 6 or 7 in as far as what we have. So at that point, if we're doing a cut and paste in on that very first row, upon the basis of that column, we're just basically saying, for this data frame that we have, on the first row, starting at column 7 through the last column that's present in the template, I just want to overwrite those contents, which are currently showing up as NA values, by providing the same equivalent section from our bead unstained intermediate that we grabbed for the last one. And then at that point, we're just correcting some metadata. We can switch around to make sure it's being called correctly, bead unstained in terms of the fluorophore, as well as bead unstained in terms of the negative. So at that point, we've spliced in the correct values for the bead unstained. You can see we have fluorophore bead unstained, negative bead unstained, and then actual values for our bead unstained here. And then similarly, we can see for like the different fluorophores, we have all their actual signature values, but we can also see that the values that are ultimately going to be extracted are on the basis of the negative bead unstained, which is also now contained within the data frame in view. So with this template now containing the detector values for both our fluorophores as well as for the unstained that we're going to need to use for the background subtraction, we can go ahead and proceed to use the background subtraction wrapper from Luciernaga, which is basically going to mediate the subtraction by matching the name that's found within the negative column to the matching unstained name that's being designated within the fluorophore column. We, of course, do also have the option to go ahead and normalize, i.e. scale the values to be between 0 and 1, as well as whether if we want to keep the unstained values present within the matrix, yes or no. So in this particular case, since we're working with beads and we're not using the unstained signature of beads within our actual unmixing controls later on in the future, we're going to set that particular argument to false. So what this is going to look like in actual practice is we take this background subtraction, we're passing it our updated template, we're setting normalize is equal to true because we're mostly interested in normalized values, and we're going to have it get rid of the bead unstained after it's been used for subtraction. And at this point, everything gets subtracted out, and we get things that look kind of on the normalized scale. We still need to go ahead and double check these, but it looks like across the board, everything has been in a relatively quick fashion. We extracted out the positive signal, and then we correctly subtracted out the background for whatever negative was being designated. So we've done quite a bit of effort. Before we go any further, we should go ahead and save this signature matrix that has been generated. Let's go ahead and save it out as a .csv file so that we have it for safekeeping and comparison. So at that point, kind of similar for any saving of a CSV file, you're designating a new location as well as the new file name. And at that point, using the base packages write.csv, You're providing it the name of your data frame, the location to store it to. In this case, since there is no row names at this point, we go ahead and set row names is equal to false in terms of arguments. And then that is saved, and you can subsequently read it back in by providing that exact same file path, and you'd still have those intermediates. So at this point, for our bead single color gating controls, through these use of these 3 or 4 different helper functions, we have basically done a lot of the automation as far as those manual steps that are being implemented back during the week 12 spectral signatures walkthrough. So let's actually visualize these initial gates to see how good or how atrocious they may be. So at that point, we can pass the entire data frame that we got back to the visualize signatures function, very similar to what we encountered back during week 12. We tell it the correct column name. We tell it we don't want to normalize it for now, just to keep them kind of like raw values because it's already been normalized. And we just want to specify the character column is going to be the fluorofluor one. And then at that point, we can play around with ggplotly. So let's go ahead and zoom out a little bit to make this easier on everybody's eyes. So as I zoom out, this is what our fully extracted panel signatures look like. Let's see if this makes sense. So, as you may remember, we had a bunch of autofluorescence left over in the APC Fire A10, as well as some tandem degradation. So let's see what it looks like in the case of the bead controls. That looks very much like I would expect to see for an APC Fire A10. And same thing, let's find our BUV 395. Let's go ahead and click. You can see, yep, that looks like a BUV 395. PerCP is always atrocious in my mind. That's why I always consider it the worst fluorofluor. And yeah, sure enough, we have all those 5 peaks on this 5 system, so yeah. So across the board, as we start clicking, we can see that for bead controls, these look very similar to what we would be seeing on like a reference library website, which means the code we use to quickly iterate through all those gate creations, place the correct placement of those positive gates, retrieve the values that's contained within those gates, and then subtract out the background and normalize it. All that little wrapper code from Luciernaga worked relatively well across the board to quickly get us to this point that we're able to see the actual fluorescent signatures for this particular data set. So all in all, not shabby, and definitely a good place to be, especially considering when I first learned how to do this back a couple years ago, I would then have to repeat rather odorous amount of code and have it either go in a for loop or multiple iterations, a lot of manual adjustments just to be able to get to this same signature view for our bead single colors. So, reasonable at first glance, we are going to see how it looks compared to the cell single color controls, which we'll get to in just a couple minutes. And before we dive into cell single colors, let me take a quick break. I'll go ahead and grab some water, and then we'll circle right back. Alright, we are now back. So, at this point, we have tackled our speed single color control gating set and correctly extracted out the signatures across the board for everybody in relatively little amount of pain. So now the beads were the relatively simple scaling set to deal with, given that they only contain bead controls, so we only had to deal with a single autofluorescence. Let's tackle a slightly more complicated use case, which is going to be more similar to what we'd see for those more complicated panels, using the cell single color controls. So in this context, let's see how this will change, especially knowing that for this particular data set, a bunch of single color controls were prepared with cord blood mononuclear cells, a bunch of them were prepared with PBMCs, and then there was both activation and non-activation conditions that we might have to factor with in terms of that negative column designating which autofluorescence should be subtracted. So once again, we can just go ahead and load in our intermediate cell single color control dating set from where we had saved it last time using the load.gs function from Flow Workspace. And then at that point, we just need to go ahead and repeat a lot of those same steps we did for the bead single color controls, as well as what we did during week 12 when we assembled everything more wholesale. We just need to make sure we update the metadata for everybody across the board so we have a good building block for that template will end up being used. So at that point, we can grab pdata, we can see what's there, we can then run the syntax that's very similar to what we see for the bead controls. So we can likely get away with just rerunning the code lines that we had using the different stringer package functions to grab out the antigen, the fluorophore, and the type from those file names. And then at that point, we have this little intermediate metadata that has antigen, fluorophore, and type. The main thing to remember is there's no bead unstained, so we don't have to deal with those NA style values. But we do still have those little bits of differences in terms of the p-Bio770 and the p-Dazzle594 in terms of the naming convention. So we'll still need to do that little mutate case when combination to be able to edit these in place, which is what we can then go ahead and do that adjustments with. And then once that is done, we can then use the Luciernaga package's detector guess to autofill in the detector column for what each individual single color's controls fluorophore should be in terms of the peak detector based on what it knows about the Aurora 5 references. And then this is going to look like. So as we can see, the little individual changes to p-Bio770 and p-Dazzle594. Once again, just because this code can be a little bit more complicated, and I want to make sure it's clear for everybody because I do find it quite useful once you start learning the syntax. You have your data frame, you're passing it through the pipe to the mutate function, and then for your column fluorophore, in the case when that column fluorophore is equal to p-Bio770 without the space, that particular row in that particular fluorophore column is going to be modified to be p-Bio770 with the space. And then likewise, if within that column fluorophore, it recognizes a value that is p-Dazzle594 without the space, it'll be modified over to be a new value p-Dazzle594 with the space. And then if it doesn't encounter these particular two variants, I want it to still keep the original values, which is why we have true and the little tilde sign to fluorophore. If this had been set to false, basically all other values would then be converted over to NAs or something like that. So with all this little corrections now done, we can assign back our new modified data frame to the p-data so we can update the metadata for our gating set. And sure enough, we have a very similar result to what we had seen with the beads, as well as what we saw with the cells during the week 12 walkthrough. And then at this point, to take advantage of all the Luciernaga helper functions that are going to let us be able to extract out the signatures, we need to generate that little template object that can then be used. So first off, we need to go ahead and generate a detector column and fill it in with the corresponding peak detectors, where we expect to see the most fluorescence visible for each of the single color controls, and then also designate a negative column based on what we expect to subtract. So let's tackle this very first one. We are creating basically those little backbones for the detector column and the negative column. For now, we can just specify cell unstained uniformly across the board for everybody. And then through this use of iterating through the detector column, matching with what's the content of the fluorophore column, we can guess out with Luciernaga what the corresponding peak detector is going to be for a Cytek Aurora 5 laser instrument, which will go ahead and all the way down fill in the detector column for us. Alternatively, if you're on an instrument that is not being guessed correctly and you end up with a few that have empty spaces, you can modify these accordingly either in your script or via a CSV file just to make sure that they're updated correctly before you proceed to signature extraction. So, in terms of negative background, this is the gating set we already were anticipating it to be a little bit more complicated, especially having gone through last walkthrough where we had to do all these adjustments for the scatter gate based on if it was a cord blood, a PBMC specimen, or an activated versus control condition. So right off the bat, we know negative background is likely to be important. So if you're working with your own data, this is really easy to do because you already know what you're doing and what you're able to do. If you're deep diving flow repository or import, you're really at the mercy of the original person who has shared the data in terms of being able to correctly match the backgrounds for each other. So in this case, SDY 3080 is pretty well documented, but I'm going to admit it's still not the best organized all in a single spot. So it's like, for example, if you looked at this lab notebook scan for this particular gating set, you're going to be squinting, you're going to be like trying to zoom in and out. But what is important is you can kind of see it is organized based on fluorophore for this particular scan, and you can see that the writing for this particular experiments, we could see which single color controls were prepared with the cord, which is showing up in the red ink. We can also see what was prepared with like the PMA activated conditions for those cases of the cytokine, and you can see there's a couple conditions where there was maybe both that were used. So in this case, you can backtrack them accordingly, but as you can see, it's going to be a little bit messy when it comes to editing this negative column so that it matches the correct background that needs to be extracted from the positive signal. So you may be wondering, is this actually going to be significant? Do you actually really need to do that? Can you just get away with subtracting out a regular autofluorescence background from the board across everybody? Well, you could try it and you could see. But it's often in conflict with that second rule for the good unmixing controls. You want to try as much as possible to make sure those backgrounds are matched. So in this case, for this walkthrough, we're going to do it. We're going to persevere just because it's going to teach us some additional skills, and we're going to make sure the negative column is correctly set for all the fluorophores so that they're matching the correct negative background that needs to be subtracted. So, what do we have to deal with? So for CD8, which is on BUV496, CD45, which is on BV510, CD56, which is on 605, PD1, which is on PVIL770, CD16, which is on APC, and CD38, which is APC 5810, as long as those viability controls seem to have all been prepared with cord blood mononuclear cells. So for those, we're going to need to change it from where it is right now as a cell unstained value in the negative column to a cord blood unstained value or some variation above being the new name designated as the background subtraction. Likewise, a few of these cell single color controls, especially those that are cytokine markers, things like CD69, TNF alpha, interferon gamma, CD107, CD25, and apparently one of these viabilities ended up being prepared with PMA ionomycin stimulated PBMCs. So these might also be not just different in background in terms of cord blood versus peripheral blood, but then activated peripheral blood on top of everything else. And then all the other ones appear to have just been prepared with unstimulated PMANC, no special variants across the board. However, as we mentioned previously, we also need to make sure we're batching the correct cell gating background when it comes to the Pacific Blue, which instead of being the regular lymphocyte gate is going to be placed on the monocytes. And then where we drew those Zombie-Ni R gates, both for the deads, would also come back into play when finding and matching the correct autofluorescence background for them respectively. So, to be able to cut down on the modifications that we're going to need in the future, let's just go ahead and modify our template to account for these different negative backgrounds using the dplyr packages combination of the mutate and case-when function. So, it is going to be long to write for this first initial one, but remember, if you're repeating a single color panel and you're doing relatively minor modifications to the protocol, this code chunk can then just sit there and not need to be modified again in the future. So this is, once again, it's a combination of mutate and case-when. In this case, instead of targeting fluorophore, we're targeting the negative column. So basically, for our negative column, in the cases when we see that the antigen is equal to the dump underscore CD14 value, we're basically going to change that corresponding row's negative value to be PBMC monocyte unstained. Similar story for those other two specified viability controls. We know that one of them was the CBMC dead unstained, so we can correspondingly change that negative rho value for it to match what we expect. Similar thing can then happen for the viability PMA. We can switch it to PBMC dead unstained at that point. And so on and so forth. You can then see for CD8, it would be, these are the regular, these were the ones that were cord bled as far as the background. We can just basically say, when the antigen column contains the value CD8, go ahead and switch its negative for that particular row over to CBMC unstained, and repeat down for these same kind of other antigens that are of this type. And last but not least, we can target the PMA stimulated PBMCs, which should all be showing up as PBMC activated unstained. So targeting each of these individual single colors as far as what is being mutated in terms of their negative column. And then we can just go ahead and keep everybody else as true, and we can set them to be PBMC unstained. So ultimately, rather complicated set of case when in mutate functions, but what we see it happens is instead of just being uniformly across the board cell unstained, everything is getting switched over. So as we can see, if we check for some of those cytokines, TNF-alpha, interferon gamma, I scroll across, what is the negative background that's being subtracted? It's going to be the PBMC activated unstained. If I check for like CD8, CD4, things that had, CD8 for sure. Where's CD8 hiding? Close, no cookie, there he is. You can see 496 has been switched over to CBMC unstained. If I check APC, which was that CD16, we can see it's a CBMC unstained. And then everybody else who wasn't being corrected ends up appearing as PBMC unstained across the board, which is what we want. So at this point, there's the three different main conditions based on the different types of original cells as well as the treatment type. So with these adjustments now having been made, we just need to go ahead and incorporate it back into the metadata, which we can do via the assignment arrow, kind of similar to what we've encountered already in the past. And because at this point we just want to make sure we don't have to do all these changes again if need be, let's just, for insurance purposes, save this also out, this data frame, as an intermediate.csv file, so we have it and can modify it worst case scenario if we need to. Which we do similar way, specify a file path, specify a file name, write it out as a CSV file. And at that point we could read it back in if we wanted to. So let's go ahead and check the data frame, just make sure there is no duplicates. So kind of similar to what we encountered to the bead metadata. There's 2 Pacific Blues that are present, and there's 2 zombie NIRs that are actually being present at this point. So both Pacific Blue and zombie NIR come up as being encountered, just like we saw with the beads. So as we check these files, we need to opt to keep the duplicate that ends up containing the brighter staining events overall, and use that for our signature. So this would of course result in, for this particular gating set after we visualized it and double checked, we'd want to keep Pacific Blue CD14, which is substantially brighter by at least a log compared to the CD19 staining. And then in case of the zombie NIR, there was a brighter set of dead cells in the context of the cord blood mononuclear cell compared to the PMA ionomycin stimulated sample. And then also there's still one of those redundant Alexa Fluor 488 controls, which was one of those stand-ins for the use of the FITC channel for the V alpha 24, G alpha 18 for the NKT. So at that point, if we're just trying to keep just a single entry per fluorophore, we could then use this filter from the dplyr package, searching the name column, removing the individual column name for these 3 ones we want to actually remove from consideration when it comes time to generate our signature matrix. And at that point, we had a 29 color fluorophores. We should have 29 rows just to make sure it's actually matching. And yeah, when we go ahead and double check, it is correct. We have 29 fluorophores, we have 29 rows. So at this point, we have all the template assembled for what we actually need to have done. We would then need to also be able to grab the events that are present within those gates, but we also want to make sure we have the correspondingly named negative gates present as well, so that we can run the template and actually have it subtract out the correct background. So let's start tackling the unstains in a couple minutes. So let's first just check to make sure for this particular gating set that we do have the correct naming syntax in terms of the gates before we let the Luciernaga package just run wild and create 29 new gates under the root or under singlets or whatever the case may be. So if we check to see what the current gate is, if we look at it, we can see we have all the way down to scattered for this particular gating set, which is kind of like what we were expecting to see, so nothing too bad as far as what we are seeing. And then at that point, we just need to go ahead and iterate through the creation of those peak detector gates based on their corresponding fluorophore specimen, just to make sure we're able to visualize across the board. So, if we're going to place this under the last of the cleanup gates, in this case it's not going to be singlets, it's going to be placed under the scatter gate. So we're going to go ahead and run this line of code. So the Luciernaga helper function in the case of initial unmixed span gate creation, we're giving it the template that we had just customized that contains the individual detectors with their corresponding fluorophores as far as the creation process. The gating set that's going to be having these new gates appended to, under which gate they're going to be drawn, in this case the scatter, and they're where we want the lower bound and then the upper bound of those span gates to be created for. And then at that point, it's going to show you the progress bar as it iterates through each individual gate creation. And then at that point, if we rerun this plot just to see how those gates look, we see that sure enough, we have the 29 new single color peak detector gates created accordingly where we expect them. And we don't have to do all this little bit of individual gate creation, all this little bit of individual customized code. It's all being handled in the background by the function itself. So at that point, all the initial peak detector gates have been created underneath the scatter gate. However, in context of the zombie R viability single color control, we ended up using the CBMC dead gate, as well as for the Pacific Blue CD14, we ended up using the monocyte gate for the process. So before proceeding, we need to double check that these still remain placed correctly and they're not accidentally on the live healthy PBMCs, in which case it would be off for what we're expecting. So we can go ahead and first verify where in our gating set are they in terms of their index value. Once again, we can basically grab metadata, pass that data frame to the mutate function, and then basically do a little intermediate standby of where is this at using the row number function just to identify the actual positions. So as you can see at this point, both of the zombies are around 31 and 32. CD14 seems to be in the 24th index position. So at that point, we know what we need to designate when we're using the flowGate package to actually visualize these and check their gates. So at this point, we want to go ahead and double check that the monocyte gate is indeed still placed on the monocytes. So referring back to our flowGate function, the geosupply gate check, we're passing it our cell single color gating set. We want to check underneath scatter. We want to check the sample number 24, see where it's being placed, and we just want to adjust the individual one, so we're going to keep adjust all set to false. And as you can see, our correctly adjusted gate we did in the last walkthrough is still in the correct position for this particular specimen, so we're all good to go. We don't have to worry about it. And then we can also go ahead and double check for our zombie R die, which was placed on the dead and dying cord blood cells. So in this case, we're modifying exact same line of code. In this case, we're just specifying the corresponding index value for that cord blood zombie specimen. And then we check it, we can see that, yep, the adjustment we had previously made in the last walkthrough is still applied. And if we extract values, it is going to be coming from that correct gate. So at this point, we have iterated through the creation of those peak detector gates. We just need to really double check to make sure that these gates were placed to what we would consider satisfactory, and if they are not placed satisfactorily, adjust them on our own individual basis based on those 4 rules that we associate with good unmixing controls. So let's go ahead and launch the helper function that will launch the Shiny app from flowGate and iterate through each of these brand new created peak detector gates just to make sure that we can adjust them accordingly before we go and extract out the values. So that's going to be check unmixing span gates in terms of the function. We're passing it our assembled template. We're giving it the gating set. And let's go ahead and say adjust all the gates as we do it across all the specimens just so they're matching each other. And ultimately what we can see is if a gate is good and we don't need to make any adjustments, as is this case for this APC R700 in the case of these cells, we can just go ahead and select done and go ahead and move on to the next gate. And then, if it's a situation where it needs some adjustments, for example, you can see this original span gate is a little bit too wide. It not only includes the bright and brighter, but it also includes all the way back to what's likely just non-specific. You can also just select span, draw a new span gate, select done, and then it should be updated and reflected accordingly. And then at that point, you just repeat for the other 27 single colors, attempting as much as possible to stick to the best practices when it comes to our unmixing controls. So here's an example of CCR4. A gate was way too wide, definitely cutting into the negative. So we could set it a little bit more narrow. It's still maybe a little bit too stretched out, but we also have very few cells here, and I don't know where antibody aggregates start and end. And then at that point, after we have gone through all the process of creating all these peak detector gates, adjusting them all to match what they're supposed to be at, it'd be horrid if we had a computer crash or something. So let's go ahead and save it as its own intermediate gating set object so that we have all these able to be reloaded in the future if we so need them to. So once again, it's going to be via the saveGS function, kind of similar to what we've seen for multiple rounds at this point. So now that our span gates have been checked, we can then do similar to what we did for beads. We can expand out the template, adding on the individual rows that will individual columns that will contain the summarized median on a per detector basis for the cells that are present within those positive gates, which is being orchestrated in terms by the retrieve average signature function. So we're still passing it that exact same template. We're passing it our same gating set. In this case, just remember, we need to set inverse that transform is equal to true. And as it's iterating, it will then go ahead and it will retrieve these individual medians on a per detector basis for the cells that were contained within these checked positive peak detector gates. Alright, and just like we started talking previously, on the bead gating set control, we needed to basically derive the bead unstained separately and then integrate it in by basically doing that little copy and paste maneuver to get it to be where it's supposed to be. So for that last example we saw, the bead gating set was already, the bead unstained was already present in the same gating set. So in this context, our cell's unstained samples are not within the cell single color gating set. We still have them placed within the cell unstained gating set. So we'll need to go ahead and load them in just so we're able to grab those background subtractions. So, first off, let's go ahead and check which unstained types we're going to need in terms of background, as we currently have it within our template. So we're taking our template, which we had called the object variable kept cells, we're pulling the contents of the negative column, and because there could be multiple entries for each of these, we just want to make sure we're keeping the unique values, so we're passing it through the pipe again to unique. So as you can see, we need to set backgrounds that are going to be specific for PBMC unstained, PBMC activated, as well as PBMC monocyte unstained. But then we also need cord blood unstained, as well as cord blood dead unstained, in terms of all the individual backgrounds that are going to get added into our template for use in the background subtraction. So if we go back and see, we need to retrieve our cell unstained gating set. So let's go ahead and grab it from its intermediate GS folder. Similar exact code, similar exact name to what we've encountered in the past walkthrough. And then at that point, if we check the metadata, we still just have a single name column, but everything else looks relatively similar across the board. So if we went back and looked at that lab notebook scan, we could eventually find there is some information that the PBMC controls seem to have been sourced from what was the ND6 specimen. The CBMCs also seem to have originated from what was the Infant 52 specimen, so basically we just need to grab these two and their activation conditions, potentially draw a monocyte and a dead cell gate depending on what we're trying to do, just so we have the same correct schema compared to what we're expecting for the background subtraction layer. So basically, let's go ahead and double check what we have. We have just the standard initial cleanup gates down to scatter. So at that point, let's go ahead and start working off of that parent gate. So if we check and see the metadata, we just want to identify where that Infant 52 and where those ND6 specimens are actually present within this gating set. And we can see Infant 52 is relatively easy. It's right on the first index, so that's not something we need to worry about. And then for that ND6 specimen, the control and the PMA seem to be in the 9th and the 10th index value accordingly. So we need to go ahead and create a couple of those additional gates for both the monocyte and the dead cells off of this background unstained sample so that we can grab the matching autofluorescence. So for the monocytes, what we can end up doing, we can basically go in here, use GSGate Interactive, create a new gate for monocytes, which is being designated by the filter ID. It's based off this first sample if we're going for cord blood monocytes. And then at that point, we're saying dimensions, keep them forward scatter, side scatter. And we want this new gate monocytes to be placed directly below the non-RBCs, which would put it as a sister gate to the regular scatter gate, which would be the PBMCs themselves. And at that point, we can go in, create it, draw it just around the monocytes. When we're happy with it, select done, and then that would be created. And then at that point, we could just go ahead and double check for the adult PBMC specimen without stimulation, how it's looking. We could go ahead and GS apply gate check. We're checking for the monocytes. We can then check for sample being 9. And as you can see, this is the original gate based off of that cord blood specimen. We can see on the adult it's a little bit brighter a little bit higher in terms of its side scatter. So we can go ahead and select polygon, do our gate check, modify it accordingly for what we need it to be, and then select done. And similar story for setting a dead and dying gate off of the CBMCs. We can just specify a new filter ID for that gate name, cord dead. We're still placing that under non-RBCs. And then we can draw our gate of interest, go ahead and select done. And then we can also double check it and modify it for that PMA atomizing condition sample, which is going to be sample number 10 based off our index values we had identified. We want to actually regate this, so we want to modify it across the board. And as you can see, we can regate us accordingly depending on what we're trying to do. And with all that done, let's go ahead and check those created gates. As you can see, we've taken that initial template. We now have PBMC dead, cord dead, monocytes, scatter. So we have all those different variants that in combination with on a per specimen basis, we could then isolate out the different backgrounds we need to actually go in and retrieve the fluorescent signatures from our template. At this point, we've made extensive modifications to the gating system. We've checked them, we've validated them. We might as well save this one as its own intermediate folder as well, just to keep things organized and kind of sequential. So we'll go ahead and save it as its own cell unstained gating set, amplified within its own particular folder. Alright, so we have the unstained specimens loaded. We have the positional gates created. All we really need to do is extract out the signatures from each of these and tidy the data to kind of match that template format that's being used for the single colors. Let's start by just extracting the medium fluorescent signature values from each of them individually. So as you can see, this is the situation for what we encountered a little bit for the bead controls. We're using the gate expers iterated function. Basically, we're targeting, in this particular case, PBMC unstained using that 9th sample, which was ND6 control. We're grabbing that unstained based off of the scatter gate. All those values are being put back into place. And then at that point, we're just changing the name to make sure instead of random fluor name, it's being correctly named in the same syntax style as what we expect to see within our template. And then at that point, we're doing it for all the different variants. So grabbing for the PBMC monocytes, it's still the same sample, but in this case, we're grabbing from the monocyte gate, so we get the monocyte background. And then for the activated condition, this is where we're going to change the sample number over, and we're going to still grab for the scatter gate. So now we have what the PBMCs for the activated condition look like. And we're just repeating down the line for all the different backgrounds we need for this more complicated subtraction schema. So PBMC dead is what we're extracting in this case. CBMCs unstained, you can see that we switched sample back to worn. And then CBMCs for the dead unstained, we're switched out the gate for the different positional one. So let's just go ahead and make our lives a little bit simpler. Let's combine all these background autofluorescences that we ended up extracting, put them all into a single data frame so they're a little bit easier to tidy up for the downstream steps. We can do this via bind rows, just like we've encountered in the past. And make sure we're not jinxing ourselves in terms of the internet. If we go to the next slide, we can see at this point we got the corresponding numeric values that we're expecting to see. And we also have, under the fluorophore column name, the individual names that we're expecting to see for the negative background subtraction. And double checking here. There we go. So at this point, since we already have them extracted, we might as well visualize the signatures just to see what kind of differences these different autofluorescence backgrounds actually have in terms of both the raw signature as well as the normalized signature. So once again, we're using the Luciernaga visualize signature function. We're generating this plots intermediate. And then at that point, we can just visualize it with Plotly's ggplotly function. So let me zoom out here so we can see them all in the same view. So this is our view of the raw unstained. You can see this is going to be the PBMC monocytes have the brightest. So this looks like 15,300 something for the MFI of the violet 7 detector. You can see this little pink guy down here is the dead CBMCs. It was only around 3,800 MFI for that same one. But then all the others kind of like are a little bit clustered around the middle. There's not very much difference. So as we started exploring a little bit last week, just because they're different in terms of the brightness doesn't mean it's a different signature. So let's go ahead and go double check to see how similar these are once we normalize and scale the signatures. We can do this in the visualized signatures by basically setting that last argument normalized is equal to true. When we end up visualizing this, once again, I need to zoom out for this particular slide. We can go here. And this is what the difference of the individual signatures look like. So if I'm comparing, let's say, cord blood versus PBMCs, if I'm just checking my PBMC and I'm checking my cord blood unstained, you can see there's a little subtle differences here and there in terms of the autofluorescence. So the adult PBMCs tend to have a little bit more of this UV signature. They're kind of evenly matched when it comes to the blue. There's little shrugs, little shoulders here and there, but nothing too dramatic across the board. By contrast, when I'm comparing the monocyte unstained, you can see my PBMCs unstained for T cells versus the PBMCs monocytes. They're relatively evenly matched. And then if I look at the activated unstains compared to the control condition, they're basically exactly overlapped. So we could have used the background from the control condition and unmixed perfectly reasonably well for what we had. So at this point, we're too far in anyway. We might as well use the correct background when we're subtracting. But yeah, there's always these little variants as far as things go. It's always just worthwhile keeping an idea of not all autofluorescence, even if it is for a T cell or a monocyte, is just going to be a single autofluorescence. You're going to have these potential variants depending on the different activation, the different types of cells, all those different conditions. So, takeaway, there's difference in brightness, but they appear to be mostly the same normalized signature. Alright, so we have the signature values for the detector columns as well as an identifying fluorophore name. We still need a little bit of data tidying before we're able to fit these values for our unstained controls into the same data frame that contains the single color controls so that we can actually orchestrate the background subtraction run. So we should go ahead and slice off the first row from the single color template, just so we know what it's actually supposed to look like. We're going to use the little slice function in this case. And as you can see, for our single colors, it's expecting this format. It's expecting a name column, a fluorophore column, an antigen column, a type column, a detector column, and a negative column. At which point it will have all the different signature columns that we are more than expecting for the individual fluorescent detectors. So basically what we should do is we currently have that individual row. We should go ahead and duplicate this to match the number of rows that are present in the unstained. This can be done kind of similar to that sequence function we've seen from base R. There's also the rep function, which is going to do the replicates. So basically saying in replicates of 1 all the way up through what we're designating here. And then at that point, we can have our little cell stand-in, which we can then go ahead and double check to see what we got. So there's two ways of going about and editing this. For this particular case, I'm doing some hard coding. Basically, I'm assuming I know which fluorophore column is ending up in which index position, and I'm just overwriting them by hand based on what I've seen, that the first column is name, second column is fluorophore, third column is antigen, type is going to be 4th, detector is going to be 5th, negative is going to be 6th. However, if your columns ended up being rearranged and you have hard-coded code like this that's going to overwrite in these positions, if type is no longer in 4, but instead is in 2, you're going to not actually have it correctly placed when it comes to this hard code. So a little caution can be taken when you're trying to slice and dice things together. So ultimately what this looks like when you get this assembled is you've basically overwritten name, you've overwritten fluorophore, you've overwritten antigen, detector, negative, because you provided little empty values across the board. You only have type still being retained correctly for what we have here. So at this point, we basically want to take this little stand-in and replace it with the actual negative values that we have extracted from the background and place them in correctly as we're doing this one here. So to cut and paste in our unstained data into the cell stand-in data frame, we need to do a few little things. So the cell stand-in starts for the detectors on the 7th column all the way to that very last column. And then for our unstained data, that first fluorescent detector starts in the 2nd column all the way to the last column. So we can just go ahead and write them into the place. And then at that point, the first column of unstained was the name of the negative background. We can just have it written into place for a couple of the metadata columns that we would expect for the single colors. So at this point in time, the correct background has been copied into place for that first part. So now UV1 through whatever R8 detector are correct. And then instead of empty particular values, you see name now has a particular value, fluorophore, and negative, corresponding to the name we had provided for that negative background across the board. So at this point, the formatting for both the unstains as well as the single colors for the cells are all matching each other. We can just go ahead and combine them together so that we can then proceed for the background subtraction. So at that point, we can use either our bind or bind rows from the dplyr package. We can combine our cell stand-in with our updated template. And then at that point, what we end up seeing is here, all our little negatives that are going to be used for the background subtraction are now in place. And then at this point, all our single colors from the cells, as far as their particular values, are also in place. So when we actually run the orchestration, basically what's going to happen is for BUV615 CCR4, it's expecting to see the PBMC unstained. So it searches the fluorophore for PBMC unstained. It sees that this is the values for the background, and then it would subtract its own fluorophore minus that particular line to get to the remainder amount. And then that same thing would be happening for each of these. So for example, for your APC, which is going to be the CBMC unstained, it would match to the CBMC unstained that's present within the fluorophore column, grab its particular values, and use it to subtract out the background from the APC fluorophore across the board. So fortunately, we don't have to do this all by hand. The helper function is going to be able to do that now that the backgrounds are present within that data frame. So at that point, we are running background subtraction. We're passing the updated template data frame that contains both the positive and the negatives all in that same data frame. We're going to set values to normalize as equal to true, and we're going to have it keep the unstains for now, just because these are cell unstains, and we may want to use those in the case of unmixing. So at that point, it will do its little run, and then we'll get back all the little extracted values that we're expecting for our fluorophores, as well as it's still kept in the unstains for visualizing their own signatures as well. And then at that point, we've put all this effort. We might as well save it as an intermediate CSV, just in case we need it later. So let's go ahead and do that. Specifying the storage location and the new file name, and then using write.csv, passing it that data frame, giving it the new stored location, and setting row.names is equal to false. And subsequently, we can then just go ahead and read that back in if we really wanted to, just to make sure it's still there. And we've already visualized all those signatures for this particular gating set in terms of the bead single color controls. Let's see what the cell single color controls end up looking like. So we can do this via visualize signatures. We're passing our data frame, we're designating the columns. It's already been normalized, so we don't need to renormalize. And then at that point, if we check to see the matrix and pass it to Plotly in ggplotly and zoom out a little bit, we can see once again APC Fire 810, as we saw last week, that is heavily APC contaminated. We still see that these cell single color controls are heavily contaminated, but then at that point we can see all the other single color controls that also make some predictions. So BUV395 doesn't look too bad. We can see 563 looks mostly clean. We can check BB510, what I'm mostly expecting. And there's a couple ones that are going to be a little bit on the weirder side. So if I check, for example, P-Dazzle, looks pretty clean across the board. Which was the other problem? Children 615 is one I've noticed has a little bit of autofluorescence contamination still present. PSI 5 I can check. You can see it looks relatively clean on this particular day. And of course, we can check what Percy PSI 5.5 looks like in the context of my cells. And compared to that relatively clean but obnoxious 5 peaks we saw in the beads, you can also see UV7, V7, and the blue tree detector are still present for this particular cell control. So as you can see at this point now, we have assembled entire signature matrices for both our beads and our cell controls. We can then compare them to each other, and then also during week 14, we can use these signature matrices or the combination of above to actually use the best fits when it comes to actually unmixing our full stain samples. So, in this walkthrough, we explored basically how to orchestrate the creation and the adjustment of peak detector gates for our single color controls. So once these gates get set up, for every fluorophore in our panel, we can quickly extract the median values for those cells that are contained within those gates, subtract out the background, and then normalize these values. These signature matrices can then be stored as CSV files that we can compare and contrast the signatures during a different walkthrough. So whether these extracted average fluorescent signatures are indeed correct, or if they will function properly in unmixing, or if we need to go back and redraw our span gates, is something that we're going to continue exploring over the next couple weeks. And with that, that's all the questions this is all I have for you guys today for this extended bonus walkthrough. In terms of remaining walkthroughs we have for this section of week 12 material, it's the more in-depth view of the Luciernaga integration function, not just for APC Fire 810, where we saw the tandem degradation, but for the other 28 variants. I'm mostly done with the writing of that one. I anticipate I'll likely record that one on Friday night. I'll keep you guys posted as far as what my plans are with that one. And then back to next week, I am back from a couple days of vacation, and we'll be back to our regular actual recording sessions for the Cytometry and R class, namely Tuesday night, Wednesday afternoon, and then Thursday morning, respectively. So, if you have no questions, I will catch you the next time.